Welcome, everyone, to the Mowat-Wilson Syndrome Foundation Meet the Researcher webinar. And thank you, Dr. Andrea Conidi, for meeting with us this evening and sharing about your story and your research. And so here we go. Thank you.
I hope you can all see my presentation, right?
Yes, can see it very good. Thank you.
Good. So I will first give some, let's say, basic knowledge of that will help us during the talk. So what is a gene? A gene is a block of information that is present in the DNA, and normally we are used to this idea that we have this DNA that is the let's say you can depict as this black line here, and then we have a bunch of information that I call here as gene A. Okay. Actually, the structure of a gene is a bit more complex, so I have to make a step back, and we have that each gene is divided in exons and introns. So the introns are the sequences of DNA that are located between the two exons. And what is far more important for the moment is that the exons. They contain all the actual information. So let's say for the proper gene, let's say the exon contain all the actual information. The introns contains let's say they form a space between the information. So they divide, let's say, they are like periods in a sentence, in a paragraph. Each exon, as I say, gives, when it's translated to a can be translated to form a protein. So we have this gene A that is composed in this case by 5 exons. And in the last exon here, we have a stop signal. And when translated, all these exons can form a protein where each exon gives a part of the protein. We know that there are different kinds of mutations that may affect the final product, so the protein. I will just focus on some simple mutations that are also interesting for Mowat-Wilson and the cases that I'm going to present later during this talk. We have that if mutation occurs in this region, for example, it can be an insertion or deletion. So one part of this exon, for example, is removed, or maybe it's added. And that will result in a shift in the sequence, and the stop signal that before was here now will be maybe a bit earlier, can also be a bit later. But in any case, you have a protein that is not corresponding to the original one. We can also have missense mutation, where a mutation occur, but actually doesn't change anything to the length of the protein. So the stop codon will always be here. And they will, but the protein will just differ a bit. So there will be something here that can be important, can be not important to the function of the protein. What's the net result? The protein can be not produced at all, can be unstable, can have a different localization within the cell, and also can have a different interaction, or can regulate different target genes. There are also another kind of mutations where there are large deletions. So in this case, bunches of the information is lost, and this most likely will not result in any protein formed. But let's go now to the main topic of this talk of tonight. Well, this evening for you, that is ZEP2 and the Mowat-Wilson. So I'll just make here a brief history of ZEP2 story. In 1998, Dr. Mowat and Dr. Wilson published this case of a patient with Hirschsprung disease, so gastrointestinal defects, macrocephaly, mental retardation, and characteristic facial features. And they found already that there was this chromosome, this part of the chromosome 2 that was involved. At the time, it was called an Hirschsprung disease with mental retardation. In 1999, in the group of Danny Höllebrook, with whom I worked in the last 15 years, a gene was cloned, it was identified, that was called CYP1, standing for Small Interacting Protein 1. CYP1 is now being called ZEP2, that is the interest for this talk. And it was recognized as a transcription factor, so a protein that can bind the DNA in specific target genes, and was identified in a screening to capture other, to find interactors for other class of proteins called SMAD proteins. It was in 2001 where the group of Masahiro Nagaya and in this paper from Wakamatsu, they found that mutations in CYP1, so ZEP2, cause a form of Hirschsprung disease that was also already presented here in the paper published by Mowat-Wilson before. And in 2002, Christian Zweyer and Anita Rauch from Germany, they proposed that actually what was initially called Hirschsprung disease with mental retardation would be now called Mowat-Wilson, because it's a totally distinct and recognizable syndrome from Hirschsprung. And in the meantime, the papers on ZEP2 started to rise. And it's amazing to see how in the years, the number of papers published with over ZEP2 and also in concomitance with Mowat-Wilson, I've increased a lot. So it was very nice, and this number is still going up and running. So it's very promising for the field. But let's go to the research part. I'm here to talk about the research, and everything started in this paper where they describe for the first time ZEP2. But what is ZEP2? ZEP2 is a transcription factor, as I said. It's encoded by 10 exons. Well, actually 9 are real coding ones. And if you see, the protein is quite big. Here, this one is the protein. These are the gene architecture. It contains 2 zinc fingers here and here that are necessary for the binding to DNA, and contains a number of other subdomains that I'm not going to describe now for sake of time. The main important thing was that in these years, we could draw some important conclusion about ZEP2. We know that ZEP2 now is important for the cell commitment. So if a cell that is undifferentiated wants to go to be differentiated, needs to express ZEP2. Without ZEP2, this stem cell here, will never go to this phase here to become a differentiated cells. We know that it's important for the differentiation, and this one is, for example, important for GABAergic interneurons, that are neurons that are present in the brain that form connections among neurons. And basically they are responsible for the formation of a proper network. And we know, and we will see later, that defects in these GABAergic interneurons due to lack of ZEP2 or low levels of ZEP2 are basically the cause of epilepsy that is often observed in Mowat-Wilson patients. We know also that on the other side, that if you have, if there is too much ZEP2, we might have cancer. So cancer cells, they express high levels of ZEP2, and this one is at the base of this process called epithelial to mesenchymal transition, where a tumor, a primary tumor, will express high levels of ZEP2. The cells will change their morphology, will become more elongated, and will be able to invade and migrate to distant tissues, so at the base of metastasis. And also we know that ZEP2 is important. You see the big screen, right? So, or because I don't know exactly what you're seeing. If you see the small screen or the big one.
We can see the your screen, if that's what you're asking. Mm-hmm.
Okay. And also we know that ZEP2 is important for the maturation of a cell. So when a cell is already almost on the way to become a neuron or whatever, still need ZEP2 to fully be functional and maturate. And this is true for neuronal cells, like in the case of oligodendrocyte Schwann cells, but also in case of immune system cells, like T cells and NK cells. So we know that if the immune system mice that lack ZEP2 or are low levels of ZEP2, they also have defects in maturation of T and NK cells. As you know, mutations in the gene, in ZEP2 gene, cause Mowat-Wilson syndrome, which is a rare disease with a typical facial gestalt, and the most distinctive trait is this uplifted earlobe, which is really a distinctive hallmark for clinicians to recognize Mowat-Wilson. Patients with Mowat-Wilson, they present intellectual disability. They can have total or partial agenesis, absence of the corpus callosum, which is the bridge between the two hemispheres. They suffer of seizures and epilepsy, and they also might have, in 2/3 of the cases, Hirschsprung disease and other defects like a delayed motoric development, heart defects, hypotonia, and also other defects that are listed in this figure here: eye defects, ear defects, heart defects, urogenital defects, and gastrointestinal defects. All of these point to a role for ZEP2 in the formation, in the proper development of the central and peripheral nervous system. We know that in the work done by Ivan Ivanovsky in the group of Livia Garavelli at the time and others, we know that now there is a correlation, genotypic genotype to phenotype correlation, where we have missense mutations that I want to show you before that are very rare, but they're normally associated with a very mild phenotype. And then you have frame shift mutations that are for the big spectrum of Mowat-Wilson patients, and their phenotype here can be from mild to severe. And then we have large gene deletions that normally associated with very severe phenotypes. In these years, different lines of research have been established to study the role of ZEP2 and the function and the expression pattern of ZEP2. Mainly, the line of research were focused on making new, different mouse models. This is something that the researcher will always do at first. First thing you want to know is how a mouse model, because it's something easy to handle, behave when the gene is totally knocked out or deleted only in specific regions of the body, of the mouse body. A lot of work was done also with stem cells, human and mouse stem cells. So cells that can become anything, but also more on the biochemical point of view. So focusing really on the ZEP2 protein per se. And it's nice of interest to see how all these line of research brought something different. Like in the case of mouse models, it allowed to study the role of ZEP2 in development, the expression pattern of ZEP2, the developmental defects associated with central nervous system, peripheral nervous system, but also like how they connect each other. Like in the case of stem cells, we know that ZEP2 is important for the exit from pluripotency, so from the pluripotent stage of stem cells to a more differentiated thing, target genes and locus conformation, but also to see how all these dynamic research, they cross each other and they help each other out in a way. So it's very nice. And also, we shouldn't forget that all this research in the end can be translated to the human condition, so the Mowat-Wilson syndrome. In this case, more than 30 ZEP2 mouse models have been established so far that allowed us to study the role of ZEP2 in different organs, like in the brain, the blood, the immune system, the eye, the liver, and so on, and at different phases of development. We know that ZEP2 knockout stem cells that help us to understand the molecular mechanism and the target genes of ZEP2 that might explain neurodevelopmental delay in humans. And we know also from biochemical studies that we know now the protein partners of ZEP2, how they cooperate to regulate target genes. So now we can have also, we can not only target ZEP2, but also other partners of ZEP2. So this is what has done in short, very short until now. But what's next? I think it's very important now to focus on 20 years have been passed, 20 years have brought very important things, but what is the next, hopefully more than 20 years in the research of ZEP2? We have now the possibility to mimic conditions like Mowat-Wilson syndrome in vitro using induced pluripotent stem cells, starting from Mowat-Wilson patients' material. We know also, and that's something that I will focus a bit later, I will go back to this later, that there are another class of mutations we should take into consideration when diagnosing for Mowat-Wilson and to find the cause of Mowat-Wilson in a patient. That mutations can also occur in regions that are non-coding, so they are non-exons. And if you do an exon sequencing, you will never find out. So we have to take into consideration also these things. I'm going to guide you through the most important thing, the first point, the first bullet point that I presented before. In 2006, there was a breakthrough in the science. It was the discovery of a system to create cells, just stem cells, starting from an adult differentiated cells. Yamanaka, who won the Nobel Prize in 2012, is the main author on this, and the system is known as, he found that a combination of factors called as Yamanaka factors are able to reprogram an adult cells, like a skin cells or a blood cell, to the state of stem cells. How does it work? We have a human donor. We can collect the cells from this donor. They can be blood, but they can also be skin cells or cells obtained during a surgery, for example, from the bone marrow. We can isolate the cells. They are called somatic cells. We can expand them in vitro. And afterwards, we can insert these 4 transcription factors, so 4 proteins, OCT4, SOX2, KLF4, and MYC, that these ones are called Yamanaka factors. And they will be reprogramming the somatic cells to a different state that we call induced pluripotent stem cells. Pluripotent because they can become whatever we want later on. So we can differentiate to different cell types, neurons, muscles, heart cells, whatever we want in vitro. Now, how this is in the context of the ZEP2 research. My idea of research, at least the idea that I applied to ZEP2, to my ZEP2 research in the last years, is depicted in this slide. So, of course, my goal was ZEP2. And I wanted to understand different aspects of ZEP2 biology. So starting from the protein, so the biochemical side, find out ZEP2 is regulated per se, find out where ZEP2 binds as a transcription factor that binds in multiple spaces in the DNA, in multiple locations on the DNA, and where it binds. And also we want to know the target genes of ZEP2. And also ultimately, we want to know what is wrong with ZEP2 during disease. So what are the genes that are misregulated due to a lack of ZEP2? And in this case, we use IPS. I'm going to show you something that I really like. It was a collaboration that we already had, my group and I had in the past with, we're still having, with Dr. Livia Garavelli from Reggio Emilia and her team. And this was also done very working together with the Italian Association for Mowat-Wilson. So we started a collaboration with Dr. Livia Garavelli, Ivan Ivanovsky, and Stefano Garafi to study different variants, different ZEP2 variants, isolated present in patients on a more, let's say, not only from the clinical side, but also in research. So what is going wrong there? How do we do this? I'm not going to present any data on this because it's still under preparation, but basically we obtain patient's material like blood, skin biopsy or urine cells. We reprogram them to IPS. We of course confirm that our IPS were fine, and then we send them to narrow differentiation. We obtain a transcriptome for these cells, and we could identify a signature, so genes that are commonly misregulated, and we know that then these genes are actually target of ZEP2, and they are deregulated. And we could then define a Mowat-Wilson gene signature, so a gene signature that is present in different patients with Mowat-Wilson. And this one will allow also to eventually in the future perform a drug screening to target these genes, either to restore their level, basically to rebalance their activity or their level in the cells. And hopefully also in the patients later, so that you can ameliorate, you cannot cure Mowat-Wilson, of course, but you can ameliorate some defects. And that's our main goal. And then I want to share something else. So in the recent years, we also been contacted by another clinician from Spain, where this clinician told us, well, I have this 8-year-old girl as a patient with Mowat-Wilson, clearly is Mowat-Wilson, but we couldn't find any mutations up to sequence. We check for other genes that are normally linked to intellectual disability, like TCR4, UB3A, and they are all normal. So, but clearly this patient has Mowat-Wilson. So we were like kind of curious, how is possible that a patient has Mowat-Wilson, but without any mutation identified? So we start reasoning, we went looking back at the structure of the gene and how is it present in the DNA. And we found basically that we thought that there might be a mutation in a region upstream of ZEP2 that maybe contains some regulatory elements that can drive the expression of ZEP2. So here is depicted here the ZEP2 locus. So here in red, you see where ZEP2 is located on the DNA in the chromosome, in the region of the chromosome 2. And you see that upstream of ZEP2 in this region here, I say upstream because the gene is translated from here to this direction. So from left to right. So that this gene is actually started from here to be translated to a protein and then goes here. In this region that is very long, it's more than 3 million bases that are it's basically empty. There are no genes. There are no coding sequences. There are nothing that can drive, not another gene. So it's just empty. And these regions also empty in the mouse and other species, and the two regions are very conserved. So this will also tell us these regions needs to be empty for a reason. So there might be some elements that might regulate that too. And now here I have to make another step to the explanation of how this works. We think now, if I always present you the DNA as a straight line, right? Of course, it's not like this. The DNA in each cell is about 2 meters long, and you cannot put 2 meters straight line into a cell. So I always, maybe because I'm Italian, I don't know, but I make always this comparison with spaghetti. So you cannot, so you have a straight, uncooked spaghetti is straight. You just take out of the box, it's straight. But then when you cook, it falls and it fits more easily on the plate, right? So, and it's also tastier. So in a cell, let's say we have a stem cell here. We have a special, in the nucleus of these cells, so here, we have a special conformation of the chromatin. So the chromatin is folded. Each of these different lines represent a chromosome. And if we zoom in, we might have a condition like this, where 2 points on the same chromosome are quite distant, and maybe 1 point B is very close to C, to another chromosome. But actually, if you would just straight this line, we would see that the 2 points, A and B, are very far, and C is another point, is another chromosome. With the differentiation, and and this one is also associated with the transcriptional levels of a specific gene. Let's say in this case, ZEP2. In this case, the ZEP2 transcription and expression is basically altered. So this conformation doesn't allow ZEP2 to be transcribed. When we have differentiation to, for example, neurons, but can be any other kind of cells, we have a different conformation. And if we zoom in, now we see that A and B are very close. So we say that they're in close proximity, even though also if we put them straight, they're still far. And C now interacts with A, doesn't interact with B. And this one, for example, for in the case of ZEP2, is associated with a green light to transcribe ZEP2, to produce ZEP2. We published a paper last year where we applied a chromatin conformation capture technique to study the ZEP2 gene, how is dynamically regulated during the differentiation of cells. So here I go back to my uncooked spaghetti and cooked spaghetti view. We found basically that if we have the ZEP2 gene here, we found that there are 3 elements that we call enhancers, because they enhance the transcription of a gene, that are located at 600,000 base pair upstream the ZEP2 gene. At the zero of differentiation, so when the cell is still a stem cell, these regions are kept aside. So the chromatin, where from ZEP2, the blue one, is tightly packed. And also this chromatin here is tightly packed, but these 2 regions are quite distant from each other, and they do not interact. However, what happened? When we differentiate the cells via a specific protocol, we have that this yellow region basically fold on top of the blue one, so on top of the ZEP2 gene. And this one is stabilized by other proteins. And these regions, these enhancers, are brought here in proximity of ZEP2 gene. And this one is sufficient to start producing the ZEP2 protein. As the differentiation proceeds, the binding gets a bit loose, but still is present. The cell becomes a neuron, and ZEP2 is able to bind to its own promoter and to regulate itself. So to conclude what we have, I go back to my picture, and I hope that I can show you, and I want to conclude with a message. So why the research is important, and why it's important to combine different aspects of the research and not only to fossilize on one aspect. So if I can say the disease, for example, yes, but if I don't know the protein-protein interactions or the target genes or how the gene is regulated, I will never have a full picture. Sorry, to conclude about this part. So I told you we had this patient, right, that didn't have, and more patients presented, then we had more patients that they were diagnosed with Mowat-Wilson, but no mute variant in the coding sequence. And we then find out that indeed we have these enhancers that are involved in the diagnosis in this Mowat-Wilson patient. So mutations occur in these enhancers that then affected the patient. So here, as I say, we can study target genes, but without knowing other information, will be just a bit of a puzzle, a piece of puzzle, not the whole picture. So all these together, combining all this together, will help us to understand the relevant target genes by ZEP2 during development and during disease. This will allow us to characterize for clinically relevant target genes that are associated with congenital defects. We can identify genes that, for example, will justify the eye defects or ear defects or the epilepsy. And this one we can then target more closely. We can have drug screenings that will allow us to restore the levels or to rebalance the levels of these target ZEP2 target genes. And then we can ameliorate the associated effects, like, as I said, epilepsy, heart problems. So this is the main goal of the research nowadays. And it's very nice that this thing is possible only in collaboration with clinicians and with family association, I think. So here I want to finish with some acknowledgements. I want to thank my group, my former boss, Danny Höllebrook, my PhD student, Judy Birkhoff, and my research technician, Anne Corporal, and all the past members of the lab, both in Leuven, in Belgium, and in Rotterdam, and our collaborators. I want to thank the Center for Biomics of the Erasmus MC that provided all the bioinformatics and the next generation sequencing. And also the most important collaboration with Livia Garavelli, Stefano, and Ivan that are really helped us a lot, and we are really happy to collaborate with. And ultimately, I would like to thank the Italian Association for Mowat-Wilson and the Mowat-Wilson Syndrome Foundation. Thanks, and I hope there are some questions and we can open the discussion.
Okay, this is Marcy, and I've put a note into the chat room. If you have a question, you can either type it into the chat room, or if it's easier, I put my cell phone number there, and you can text it to my number, and I'll ask. But I have a couple of questions, Dr. Conidi. So in the research that's being done on Mowat-Wilson syndrome and the ZEB2 gene, there are actually studies done using mice, but then also studies done using human cells and tissues from biorepositories. So how important is it that those biorepositories exist for researchers to use?
They're fundamental for us. Just a quick answer is they're fundamental. So we collaborate a lot with a biobank, a biorepository from Italy. For us, it's so easy to access the material. Of course, we don't have as researchers, we are not allowed to know details of the patient per se. So all the material, of course, is anonymized. But yeah, we can have some basic information like the age or the sex or the gender of the patient that are important for us for other things to take into consideration, like if you want to sequence or compare the stuff. But I must say, in my experience, working with the repositories made life much more easier for us. So we could easily get the material very fast. We could access to different kind of sources like blood cells or DNA, because sometimes we just want to have the DNA of the patient. So it was very nice to work with. So it's really fundamental. It's fundamental that these repositories are well kept, are well maintained, and of course it takes time and a lot of effort, but they are
Right. So I'm not sure if you're aware, but I want to let everyone know that the Mowat-Wilson Syndrome Foundation is currently in the final stages of revamping the patient registry, so where just data and information can be kept and families and individuals can send that information. And then our next, of course, important thing to do is going to be working on such a biorepository as you had access to there in Italy. So that is something we certainly look to do to help support researchers. So we have a question. I'm going to read it. A quick question. Your research will allow patients with Mowat-Wilson regulate the missing program ZEB2 for them to be able to differentiate stem cells and repair maybe their brain connections. So I think what this question is asking is kind of what the hope is in the future, that as we get to know, or researchers get to know more about, and it from what I understood you saying, it's not anymore just about the ZEB2 gene in isolation, but all of the things around that too. So reaching out and doing that in hopes that something could be done in the future. Can you maybe speak a little bit about like what your dream of dreams would be in the future that would come from all the ZEB2 research?
Unfortunately, any neurodevelopmental syndrome, and not only Mowat-Wilson, but like also Pitt Hopkins or Angelman syndrome or other kind of neurodevelopmental syndromes, they cannot be cured 100%. So we can restore eventually in vitro ZEB2, but then this would mean to re-inject these cells in the patients. And even if you would reject these cells in the patients, we cannot restore the brain connections. So we can work at the level of the target genes, some genes that we can, for which we can find some compounds that can restore the levels, but we cannot really restore the levels of ZEB2 or repair, let's say, recreate a corpus callosum, or so this is the unfortunately, it's something that we will never be able to do. We can, as I said, find new drug targets, new compound, maybe that can ameliorate something, some other thing. And also this also goes linked to the second question, whether they
would there be a type of pill or injection? Yeah,
so this is not fortunately, no, this is not something that we can do. I say we can more.
So in essence, what you feel the future may hold will be some type of therapy that would maybe reduce some of the conditions surrounding Mowat-Wilson syndrome. But is that correct?
Yeah. So I once had the opportunity to talk with families, and for example, I make an example. So I was talking with these families, and for most of them, epilepsy was the main issue. So it's something, of course, it's scary, and there are a lot of therapies, and of course, you want to yeah, you want to keep it under control, right? Now, for example, we found in our studies a number of genes that are involved in epilepsy. So that when the levels of these genes, of these proteins that are regulated by ZEP2 are up or down, you might have a bit more epilepsy. Of course, then this is to focus the research not anymore on ZEP2 per se, but on these other proteins, and to find something that maybe can block or can activate these proteins in neurons, and then you can restore partially the function of the cell. And if you do to a level of the brain, then you might help ameliorate the epilepsy. That's, let's say, my my personal goal is that, to be honest. So yeah,
Right, right.
I
And
really always,
and I appreciate that. I've spoken to Dr. Mowat, and is kind of what he was saying. We can't, there's nothing on the horizon that would indicate that there's going to be any type of cure for Mowat-Wilson. However, with all of the research being done, there might be some types of genetic therapy and other therapies that can be done to help lessen the severity of some of the things that come with Mowat-Wilson syndrome, and which could also lead to longer lives and just better quality of life for
yeah, it's
people with
also not only for the patient but also for the parents, and
yes.
that the management of the patient per se is also another let's say interesting aspects. That, and it's nice to see how with my eye how the research is more and more crossing the clinicians, but also the family life and the patient's life. So it's very nice to see this thing.
Yes, well, that's one reason we wanted to, this is our first Meet the Researcher. We feel it's very important for families to understand why things like the patient registry and biorepositories are so important. And as you noted in your presentation, there has been a great increase in the number of studies on Mowat-Wilson syndrome and the ZEB2 gene. And we certainly applaud you and your efforts. I do have to ask, though, real quick, and a little more, less of the researchy, I guess, kind of question, but what was it that, that at one point in time made you want to say, this is what I want to study?
Yeah, in my case, personally, I come from a background where I started as studying breast cancer, human breast cancers, and yeah, at a certain point it became personally too much, because in that case, we were working with patients' tissue, and sometimes, yeah, when you ask for a follow-up, maybe this patient is dead, or so. It was at the point I needed a bit of distance from this. So I started doing my PhD in more biochemical environment, more biochemical studies. But then I always miss a bit this sense of, being useful, to actually being useful. And I was working on ZEP2, so yeah, for me it was always very, very interesting to see how can we actually translate our findings. I mean, as I said before, we can have all the findings in this world in the research, but if we are just keep it in the lab, then it's a bit useless, I think. And so I wanted to translate these, and then I had really honestly the super chance to interact with Livia Garavelli and her team. I would never stop thanking her and her team, because they are just amazing, and the interaction with them really gave me a boost, and I like, okay, really, this is a field where I feel that we can be useful as researchers, and we should be useful. So that's
well, we're certainly appreciative all of your wonderful efforts. And we appreciate you coming here this evening. First researcher at our Meet the Researcher. And for those of you that are here this evening, please stay tuned because we have another upcoming webinar in October, and I'll be sending that information out. But we do have one kind of last question, and I know this is a tough one, but when we talk about the possibilities and the future and what things could be done perhaps that research kind of promote, and as we better understand the ZEB2 gene and all of the things around it, and think about of therapies in the future. I mean, are we looking 50 years out? Do you think we're looking 20 years out? Is there just no way to know at all?
It's difficult to say now. I mean, consider, I just make a very small example. If we differentiate cells from patients to neurons, it might take also 100 days to differentiate, to have full functional neurons, and sufficient eventually to perform any drug screening in a sufficient number. So you see that already now we're still talking about very long time scale, 100 days, a third of a year. So it's very difficult to predict if it's 10 years, 50, 20, or 5 or 2. You never know. So it's
right. So what's important is for us to keep supporting research and for us to keep more. And
I think it's also important for researchers to interact more with the kind of foundations or family associations like yours.
Right. And so we really appreciate you coming. We really appreciate all the people who have attended today, reaching out. Thank you again so much for taking the time to speak with us. I know it's early morning for you, and you have your family, and you have a young one of your own. And
yeah.
So we say thank you so very much for sharing your research. And again, we just appreciate your efforts in the research, and are very grateful that things worked out to where you were able to connect with all of these organizations and hospitals and foundations to help support your work. And we look forward to hearing more about what you're doing, and perhaps supporting you in some way in the future. So thank you very much. And again, thank you everyone for attending. Look forward to seeing you in October next webinar series, and I'll be sending you out more information about that. So everyone, have a lovely Labor Day weekend. Thank you.
Yeah. Thank you. Bye.
Thank you.
Thank you for attending.